Okay. Okay.
Well, so it's a pleasure to have Petra today here. So it's a very good friend, and I should say that, well, I don't remember when first I met Petra. So, but we met several times during these several years and a lot of interests in common. So it's a person that I really can say that I have been following his work, not only as an eventual collaborator, but as someone who have worked in subjects that I'm interested on. But more than that, Petra is, I think he got his PhD in the early 90s in Czech Republic, and then started an independent career that was mixed with visits and periods of postdoctoral sabbatical leaves in several places. One that we also had in common, that was the Arizona Insect Science Center, where he collaborated with John Law. That was a kind of key person in the insect biochemistry scenario, and also interested in iron and things like that. So, well, Petra is a person that has worked in several related subjects with a few things in common. So most of his, although he started with Galeria, most of his career has been with ticks. And in ticks that are a kind of a model that I should say that much less people work with ticks than with insects. So then the molecular background, the genetic background is much smaller. So people have to work to get all concepts from insects, which sometimes helps, but sometimes does not help because it's a different animal. And there are a lot of kind of concepts that in tick biology that not always have a very strong experimental evidence, others have, but of course. But Petra has been visiting several key aspects of biology of ticks, like digestion of blood, immunity, and immune protein, immune reaction, interaction with parasites, heme biology, iron biology, so gene expression. So, and in all those areas, it's interesting that he has not only made good work, he has in providing seminal contributions to the field, saying that, well, then, and these are the enzymes, these are how the pathway works, this is these are immune proteins, and this is not only solid and very straightforward experimental designs, but also very good and solid and technically sound approach. So I should say that it's a type of paper that I usually say I like to read. So it's something that you say, oh, okay, this and that, and this is opinion, this is fact, this is confirmed. It's everything clear, well written, and well designed. And so it's really always a pleasure to read his papers. And it's Of course, therefore, a pleasure to have you here, Petr. Thank you very much for accepting that invitation.
Pedro, I feel a bit, yeah. So, thank you for these nice words. To be honest, we very much follow the Brazilian science and TIC research, and are very inspired by your work and others. And we would like to copy, or sometimes we will see in my presentation we are following your ideas, and yeah, and I really like the science which is done at your side, and very much appreciate the cooperation we made with Jose Marcos Ribeiro. And I'm afraid my presentation will be a little bit not going too much into the details. I leave aside completely the TIC immunology, because, and I'll try to keep more about the things we have more in common. So I call it Hit the TIC, because our effort is somehow to find it's kind of cliche, but I like to say it, find the Achilles heels in TIC physiology and try the way how to target these vulnerable targets. So I go ahead, I guess.
Yeah, just one small thing before you really start. We most of the time suggest to people to make questions during the presentation. So because this is the type of thing that this media allows, so people to ask things, because frequently when you let it to the end, you don't remember what were the questions. So then you have more questions, people can stop, and you are at your place, at your desk. I'm here at home at my small veranda, and then so drinking a beer. Then just I will making a question, and if other people also have questions, please do. We have time. We don't have a fixed limit, so it's easy to. Okay. So.
So first of all, I'd like to briefly introduce the TIC research team in Budweis, in České Budějovice. So we are actually quite a lot of people around, and we are searching in all the three parts of the TIC pathogen host triangle. Now it's even coming to the fourth corner, trying to involve the story of microbiome interaction with the TIC and pathogens. So my lab and Ondra Hajdušek and Radek Šíma lab are now joined. So we are actually one. Can you see the mice, or shall I put the?
Yes.
There should be some. Laser. So these labs are now joined in one, merged, and the new boss, it's a young scientist, Jan Perner. So he took the lead, because directors think we need to change a little bit and go, and he's a very talented guy. So I think he will do excellent job. So we are more on the TIC pathogen interface. There is a large group led by Director Libor Grubofer and now by Ryan Rego. So these people are focused on tick-borne pathogens, and I would emphasize especially the laboratory of virologists led by Daniel Růžek. They are doing now excellent science, not only about tick-borne encephalitis, but they recently published wonderful paper about the coronavirus, and they are really doing very well. There is, as you know, Michalis Kotsifakis, Michalis is mainly focused on salivary proteins, interaction between tick and the host. And at the faculty, which is actually within the same campus, there is a laboratory which is very much focused on host immunity against ticks, so molecules like yeah, how the host react, usually having mice as a model. So yeah, I just now like to introduce the key person in our tick research, which is Jan Erhard. So he's taking care of our tick colonies and doing it very well. And I think we are all completely dependent on this smart guy. So I think you already know this because I showed it several times. So in my opinion, there is no chance to find some common strategy to fight, to combat ticks, and we need to think about it. Let's say for human, we need the best would be to be well aware of a tick bite. For dogs, we need something which doesn't smell, and it's repellent or the chemicals which are very well protecting for cattle. I think there is still nothing better than the BM86. So I guess I have some. So maybe you noticed this recent paper coming from Errol Fickrick lab. So they used the mix of I think 19 mRNAs as a vaccine encoding the salivary proteins, and what they actually achieved by this is they enhanced the resistance to the tick bite. So when they see the immediate inflammational response and remove the ticks, they certainly prevented the transmission of Lyme disease. So it was published in very nice journal quite recently and get a lot of public interest. For ticks, for dogs and pets, I think we have the things like this, especially the generation of the isoxazolines like Bravecto based on fluranal, or it's very promising. And these chewable tablets, I use it also for our dog. And of course, the BM86, and people are still trying to get something more universal and better. And I don't know if there is any, if you are aware of any efforts to find something against the reservoir animals. I'm not aware that it should be useful strategy. So our model is Ixodes ricinus in Europe, so very close relative of US Lyme disease vectors, Ixodes scapularis, and so it transmits mainly the Lyme disease in Europe, and also the tick-borne encephalitis. And we have some now good tools to study this. So the great advantage for us was the availability, the knowledge of the genome of Ixodes scapularis, which is still improving. Then mainly thanks Jose Marcos Ribeiro, there are several very nice and useful tissue transcriptome from this species. Of course, for functional study, we have the RNA interference. Even though now there are some reports that people are trying the CRISPR-Cas. I'm not sure how this will be usable for ticks, but at least it's interesting. And what we have now enhanced is also the transmission model for Borrelia and Babesia in lab, and quite recently, and we introduced the in vitro membrane feeding, which really allowed us to make some progress in the basic research of tick physiology. So Petr already mentioned this guy, so I'm very grateful because it was actually him when I came for a short stay to to University of Arizona, and John asked me this question: what do ticks do with all that iron? He was very much in this, research of ferritin and transferrin and this stuff in manduka and other mosquitoes, and so. And during that time, I started work on the tick ferritin in 2 species. And since then, we also started to be interested what happens with all the hemoglobin and iron in this heme. So here is a short overview. So one tick, one adult female take like 1 milliliter of blood, which contains maybe 150 milligrams of hemoglobin, and out of this is 6 milligrams of heme. And if this heme will be cleaved by heme oxygenase, it will produce half milligram of iron. And the original weight of the tick female is 2 milligrams. So theoretically, it's incredible dose of potentially pro-oxidative compounds. So ticks definitely have to adapt how to deal with that problem. So that was something like the start of our interest, and there are some some physiological processes associated with blood meal processing and actually this extreme gluttony of ticks. So we focus on the digestive cells, trying to find how they uptake the nutrient, then mainly about the blood digestion, then also what happens to heme and iron, then a little bit looking at ovaries, but I think the Carlos Lugo and other people in Brazil knows much better than we do. Also what happens in midgut lumen and how the heme and iron is how actually distributed inter tissues. So this is kind of overview picture which a little bit define our interest. So at the very beginning, we focused actually at this stage of the Ixodes ricinus. The reason was quite practical because we were able to reliably dissect this, get tissues, get midgut, get salivary glands of this, which is much easier than get it from the full fat ticks or some smaller ticks. So, and when we see where to target the ticks, so for pathogen transmission, we should target it at the very beginning of the tick attachment. For further development, we should somehow interfere with rapid engorgement, and for reproduction, we should somehow interfere with actually egg production. So this is basic strategies. So this is a very old figure from review written by Dan Soika and us. So actually showing what happens in tick digestive cells. So there is hemolysis of the red blood cells. There are some information, one from Jose Ribeiro and some from Japanese author, that this process should be enzymatic, probably by some serine proteases, or we think now about it, also some lipases can be involved. Interesting feature in the tick midgut, that in blood, the hemoglobin from the blood from some species like I think guinea pigs forms wonderful hemoglobin crystal. Then the hemoglobin is uptaken to the digest cell, most likely by the receptor mediated endocytosis, and the other proteins goes maybe by food phase. fluid phase endocytosis. Yeah. This is a rough scheme. So we focused on enzymes involved in hemoglobin digestion, and later also on the albumin digestion. So hemoglobin, as I mentioned, goes most likely via a hemoglobin receptor, which to our knowledge has not been identified as clearly. So, but it's believed mainly from work made by Flaviola in a Pedro's lab that it's hemoglobin receptor mediated, and the hemoglobin goes to the large endosomes, and most of the hemoglobin digested, most of the heme is detoxified, going to the actually the hemosomes, the small particles full of heme. So the majority of heme from the blood, digested blood, is detoxified by this. And the other proteins like albumin, they go to the small endosomal vesicles. And the portfolio of enzymes involved in this is basically the same. So it's based on acidic catepsin, mainly of papain type, like catepsin L, B. In hemoglobin digestion, there is probably involved the catepsin D, which is aspartic peptidase, and then it's down cleaved to the exam with the exopeptide activities of catepsin BC, and there are some serine proteases probably, and metalloproteases involved in digestion to free amino acids. And the same portfolio was of the same network of the enzymes seems to work also in these small for digestion of other proteins, other serum proteins, not hemoglobin. Still, a lot of questions remains to be investigated. So we know almost nothing whether the same enzymes are working in other developmental stages like larvae or nymphs. So this is one question mark, how it is here. And another big question mark is actually the major digestion which occurs in fully fed ticks after they drop off the host and they use all the blood for making the huge egg clutches. So this also needs to be investigated. So this is what we are trying to understand now. So the current ongoing project of one PhD student, Terka Kozelková, so she focused on proteomic analysis through the development. So she is doing the proteomics analysis of nymphal midguts, unfed, 2 days fed, fully fed, and then these nymphs which actually are during the stage of digestion and molting to the adults. So she got some interesting results. Luckily, we have a lot of information from the transcriptome done by Jan Perner with Jose Ribeiro and others. So there is a lot of we can rely on and use for the. So I think this can be published soon, at least some basic overview of the enzymes. And what we also noticed and also attracted our attention that in the transcriptome we did with Jose, Marcus, and Perno, that we see that there are some lipases very highly upregulated at the very beginning of feeding. So they go up in 3, 4 days and then drop down again again in the fully engorged ticks. So we now try to enter the new field, which is actually a lipolytic system of ticks, which is very much, I think, unexplored yet.
Petra, may I ask you a question?
Yeah.
If those lipids, of course, when we see hydrolytic lipase enzymes, we always think that if you are dealing with a gut, that they are digested. But sphingomyelin, there are not so much sphingolipids in blood. On the other hand, if just thinking of something weird, if you are thinking of a tissue that needs a lot of membranes, there is, I think the people in Chagas disease did it. There is some work on the that shows that lysosomal sphingomyelinase, the acid one, works as a signal that induces membrane turnover. So maybe this is not to digest blood, but to act in a recycle of membranes that are being used for endocytosis.
I see. Yeah. That's the lipid metabolism is incredible. So what I show here, so it's this is the stain for the lipid droplets. So these are the midguts from nymph after being molted from fat larvae. So we see in 1 month it's full of lipids, and still half an year after, and actually it's become get rid of the lipids after 2 years. So it's really like lipid storage. And what Veronica just showed me today, that actually when she fed these 2 years completely starved nymphs, start for 2 years, and when she fed them again, so the lipid droplets appear already in 6 hours. So there should be some very, very fast also lipid synthesis. So this we will also focus on what can be behind such a really potent and lipid synthesis, and this metabolism is really fascinating. This is just for illustration, a nice picture of this 1 month old nymph, and next to it is the synganglion, and this is showing the 1 year old female still full of lipids. Actually, even 2 years old females, they still have enough lipids. So it's kind of, we suppose that there should be some lipases really needed for either getting the lipids from host blood at the very early stage, and then should be some lipases actually for the long-term starvation of ticks to get the energy for long periods of starvation. So we tried also to a little bit to touch the question about the nutrient sensing. Maybe not that good as Carlos Lugolor did it in his work, but yeah, we found in a tick megatranscriptome the insulin receptor. So we try to target this insulin receptor, found not very strong, but still significant impairment of feeding when we silence it by double-stranded RNA. And we also try to do the same for the kinases like TOR and ACT. And I think this was confirmed by other authors, that actually the act, silencing the act, shows the nice phenotype of really impairing tick feeding. So it may be that the act would be good druggable target. We tried some drugs for ACT in using our artificial feeding, but we actually didn't see, but not sure whether these inhibitors are stable enough in our system.
So, sorry, I lost something. This is the weight measured when? How much time after feeding or after injection? I lost the.
I think this is the day for the fully engorged ticks, I guess. Yes, because of the weights these weights are for the fully engorged females. So like the day 7, day 8.
Okay.
And now we are also trying to characterize, because in Ixodes scapularis and the same, there are clear orthologs in Ixodes ricinus. There are 4 insulin-like peptides. We named as like 1, 3, 4, 5, because the 2 actually was not confirmed to be insulin-like peptides, but it's based on the paper published by Gulianos. So most of these insulin-like peptides are expressed in ovaries, like 1, 3, 4, 5, but we are mainly in our focus is now the insulin-like peptide 4, which is expressed in midguts and salivary glands, but not in the other tissues. So we think that this might be somehow involved in nutrition sensing for the guts. So I mentioned the membrane, artificial membrane feeding.
So it has several insulin-like peptides. So, but those should go to the hemocell, and then every tissue should respond to them.
Yeah.
To them all. So what's the difference between them? You think they should target different receptors, or this is a time or something? So
you are exactly right. I think they target the same receptor on because we found only one insulin receptor in all tissues. There are no, so we think that the specificity, tissue specificity and responses, it's based on these on the insulin-like peptides. So that we would like to get closer to this point, looking at the candidate tissue for this is probably the synganglion. But in producing these things, or maybe if we will be lucky enough to with the proteomic sensitivity to see these insulin-like peptides in hemolymph, as you suggest. But we would expect the specificity in tissues for, actually driven by these 4 insulin-like peptides.
Mm-hmm.
So this is just the picture of the inventors of, maybe not the inventors, but the people who really started to make artificial membrane feeding of Ixodes ricinus. And Jan Perner was sent to them to Neuchâtel in Switzerland and learned well the technique. And after coming back, we could start to solve the question, which was actually open in Petr's lab by Gloria Brass, if I'm not mistaken. They biochemically found that ticks are not capable to synthesize heme. So the genomic and transcriptomic data actually show that pigs lacks most of the enzymes in heme biosynthesis, and specifically exotic species, they maintain only these three mitochondrial enzymes, and we still do not know. They should be functional, otherwise they wouldn't keep them, but we still don't know in what processes. So it's one question for further research. And other interesting and very important thing for in tick is that they apparently lack the heme oxygenase, the key enzyme which is needed actually to recycle iron from digested cleaved heme, from the cleaved porphyrin ring to release the iron and get it back to the circulation and to the system. So these are the features very specific for ticks, even though the heme oxygenase is apparently missing in some mice. So based on this, we saw that might be good target to focus on these really different departing traits from the host. So this is work done by Jan Perner. So he was able to feed ticks either on full blood or on serum, and what he saw that the serum fed ticks are still quite happy, laying this comparable amount of number of eggs, but there is no actually embryo development in these eggs. So actually they show that the ticks need maternal heme from hemoglobin for its their progeny. And wait here, but a little bit discouraging is that when he supplied maybe 100 only this amount of physiological concentration of hemoglobin, it was still enough to getting the larvae. So the chance that we will be able somehow to impair the heme distribution and to such a level that there will be no progeny, it's actually not very promising. So you see that when we fed with 1% of hemoglobin, there is still a lot of heme in eggs, and so maybe we just things do not have that much troubles to get rid of the excessive heme via the hemosomes. So Matej Kučera, our other PhD students, which are unfortunately now leave the lab, he was able to get the full cycle of ticks fed either or on a serum, fed on full blood, and certainly so the technical problem was finding to the thickness of the membrane, so they can get the mouse part of the larvae, let's say, to get feed. And yeah, and the message out of this is that actually the amount of heme the larva acquires from the eggs is actually enough for the whole cycle till adult fully engorged females. And this is the work done. I think it was very important transcriptomic project performed with Jose Marcos Ribeiro. So we focus on mid-guts of 3 days fed or fully engorged ticks, or fed on a full blood, or fed on serum, and the basic question for the transcriptome was what is actually upregulated, what is upregulated or downregulated by the presence of hemoglobin in the diet. And to our surprise, there were not many proteins clearly upregulated by the uptake of hemoglobin or heme. One of these proteins we characterized was glutathione S transferase of delta epsilon type, and interestingly, it's this. I mean, TIC has mainly 40 genes encoding different glutathione S transferases of different classes. But this is very specific, this one for ticks. We didn't find it in any other species, and this seems to be really specialized for dealing with heme. It's upregulated by the presence of heme or hemine in the diet, and we believe this enzyme is involved in intracellular detoxification of the heme in digestive cells. Another recently characterized proteins.
Petr, sorry, I missed how a glutathione S transferase can metabolize heme.
Hannes, please say it again. I missed the question. How
A glutathione S transferase, a GST,
yeah,
can metabolize heme.
I think it doesn't metabolize. It just works like the most of the glutathione transferases work like the detoxification enzyme of different things. So it's just it doesn't metabolize heme. I think it's just maybe transfer it to somewhere, but I don't think there is any cleavage of heme by this enzyme.
So you think there is some conjugation of heme on some other molecule, or?
Yes, or maybe it's somewhere in between the digestive vesicles and hemosomes. So another detoxification enzyme was described by Flavio. This is the ABC transporter, right? So we think this works in a similar way, but not exactly know where it goes and where it transfers to heme to what is the tissue, or whether hemosomes are actually taking this heme from glutathione transferase. We only know that it's really upregulated by the presence of heme, and it's binding heme. So it's heme binder, but it doesn't metabolize it, I guess. Yeah. And what happens? Maybe Pedro knows.
May I just jump? There is an old paper, I don't remember, it's someone from Israel. I don't remember the name now, but that published something showing that with glutathione transferase purified with heme, just the heme disappeared. It was something like the color was vanishing. So, but they did not made any kind of more deeper study of what is happening chemically. So, but it was something like that, and that nobody never did anything with that, not even saying that it was wrong, or making or following that path. But those things are there. In Hodgkin's, the kissing bug, we showed that there is a heme degradation path that starts with glutathione, adding a glutathione, and we never found the enzyme, but it could be a glutathione S transferase. But in that case, we found a derivative, a kind of heme biliverdin conjugate And in the tick, we never found anything green. So then it's a kind of weird. So there was not, we never found anything that could be an open porphyrin ring or something like that. Or so.
We got the same results trying to find the biliverdin or actually the metabolic products of heme. So we didn't find it in the study made in 2016. So, but this is really interesting. So I think we have to look at this. It's very interesting, and maybe we are on the track on the same effect. But what happens to the heme further, we don't know. I mean, bound to GST. So in a
Also, can I add a
comment.
question? That it's Clarissa here.
yeah, Clarissa.
Yeah. So I was looking at the images of the ticks that developed completely without the hemoglobin. What happens if you add hemoglobin in the middle of development or in the late? Is this adaptation to have low heme in the diet is permanent, or they rapidly change the expression to adapt to more heme, or if they die, if they get heme suddenly after development?
I think when we add the hemoglobin during the feeding, sometime the third further we will rescue the phenotype. So
they adapt again easily.
Yeah, I guess. Yeah.
Jose here, can I make a comment?
Yeah.
Yeah. In your previous slide, you showed that there is a one-to-one binding of the heme to the glutathione transferase. Right? You see that molar ratio, when the molar ratio is one, could it be that this is working just like Pedro's hemoglobin binding protein that, not the hemoglobin, the heme binding protein that was in the hemolymph? My question is whether when this heme is bound to the glutathione transferase, could it be that its pro-oxidative nature is inhibited?
Mm-hmm.
This was shown, we did that with Pedro heme binding protein. So that's my question.
Yeah, well, we think it actually works like the intracellular mop for excessive heme, but
is the concentration
we were able to silence this and didn't see any strong phenotype by getting rid of this enzyme. So
is the concentration of this enzyme high in the hemolymph?
a good question. I don't know whether
Because if it would have this binding activity, it should be higher than the free, just like in rodents, the concentration is also quite high.
But it will not be in a hemolymph, or if I misheard you, it will be within the digestive cells, intracellular.
Okay.
In the hemolymph, there's really the help, the main binder.
So this would be a more biophysical than biochemical role.
Maybe a transporter.
Transporter, maybe.
A transporter or an oxidative protector.
Yeah, but the point, one point that's missing is that you have digestion is into a lysosomal-like a vesicle, and the heme granule, the heme platelet, that's the hemosome, is another thing. So the heme should go through the cytoplasm to another organelle. That
Alright.
may be a good candidate role for the GST.
That's what we suppose, that it's somewhere on the way out in the cytosol, doing the kind of binding and protecting against oxidative stress in cytosol, but
And
the
I
phenotype was not
learned,
that strong as we would expect.
I learned that in plants, at least, there are enzymes that conjugate molecules to glutathione and put them then into vesicles through ABC transporters. So
Mhm.
that starts to be very close to the model that Pedro suggested.
Mhm, mhm. Okay. Oh, you got still time? Carlos told me for one. So
Don't worry. Don't worry.
okay, good.
Yeah.
That's
Okay.
my discussion. So the other protein which is involved in heme, intracellular heme transport is this actually not ortholog but related to the so-called heme responsive genes, first characterized in cellular haptitis in nematodes. So Jan Perner found its ortholog, and since the simple RNA knockdown didn't show any clear phenotype, so he used the yeast, the model of yeast, which is actually not capable to synthesize heme because it lacks the first enzyme on this aminolevulinic synthetase. Am I right? Sorry. So this need to grow, to get the disease grow, they need to be supplied either with this aminolevulinic acid or heme. So he used this model to live and compare it with what the other guys in Spain did with the heme response gene from Leishmania, and using these growth experiment of other disease, he was able to show that this HRG actually acts as the heme transporter or exporter, and the additional experiment he used the toxicity of heme toxic analog, the gallium protoporphyrin. So when the heme transporter hit the heme responsive gene was silenced, so it protect against the toxicity of with the gallium protoporphyrin. So this was another evidence that this is really involved in heme intracellular transport. Regarding the intertissue distribution of heme, so there are two main transporter in ticks. One described by Clarissa, if I remember well. It was called HELP for heme binding or hemolipoglycoproteins, which are very close to the insect apolyphorin, and in other tick species is called carrier protein. So these are the most abundant proteins in tick hemolymph in all stages, but other heme binder and the more important for transporting heme from to ovary and to eggs, it's vitellogenin. So there are in Exodus at least, there are 2 vitellogenins, and by sequence or structure, these helps and vitellogenins are very much similar. So it's very difficult to say just based on sequence that what is vitellogenin and what is carrier protein. In most transcriptome, the helps are annotated as vitellogenins, but there are vitellogenins are only expressed in fully fed females, and there are many expressed in gut and trachea with fat body and salivary glands, but not in ovaries. They are transported to ovaries. And here are some knockdown experiments. And so here we can see that silencing of the help actually doesn't disturb much the transfer of heme to the ovaries. The main effect was achieved when we silenced the vitellogenin 1. A little bit less in vitellogenin 2. And the question which remains to be solved for us, maybe you know, but we don't know, these vitellogenin 1 and vitellogenin 2, whether they work like a heteropolymer, together into transporting, or only one is involved in heme transport. This should be, and we like to get good student to solve this question. Good is that we have a very good antibodies again, and really specific for vitellogenin 1 and vitellogenin 2. So maybe if we have a student who would be like to come to us and try to help us to solve this question, it will be very interesting. Kind of, I hope, not very long project. So as I mentioned, there is no heme oxygenase. So one question was where the iron actually needed for the physiological, for the metabolic enzymes of the comes from. So we got some I think good evidence that the main source of iron is actually the serum transferrin, which is digested in acidic environment of the small endosomal vesicles. So here are some experiments. We see that actually we can monitor in other tissues the iron level on the expression, not expression, but translation of ferritin 1. So you see that there is no difference whether we feed on blood or serum. Maybe even serum provides more iron and more ferritin in tissues. And when we edit, simply edit, maybe multiply when we doubled the dose of the physiological amount of transferrin, just adding the transferrin to the serum, we can see that there is high increase in ferritin 1 in the ferritin 1. So we think that transferrin is main source of iron for ticks, and this is what Fanny knows very well already from the old dissertation of Ondra Hajdušek. So there are 2 ferritins in ticks. I think in most tick species there were found two different ferritin. They differ in that so-called ferritin 1 is intracellular. It doesn't have the signal peptide, but it's regulated using the iron responsive protein. So when this iron responsive protein is actually silenced, the ferritin expression goes up. And there is another ferritin 2, which is secreted to the hemolymph. It's mainly present in hemolymph. It's synthesized in gut, and when we silence this, we see that the message or the level of ferritin 1 as the acceptor of the iron is actually lowered. So our interpretation was that this ferritin one serves as an iron intertissue iron transporter. And interesting for these ferritins, both 1 and 2, in most mammalians and insects, the ferritins are actually heterooligomers composed of heavy type and light type chain ferritins, and this thick ferritin seems to be kind of hybrid. They contain both the signature domain like the peroxidase center of heavy type and the nucleation center of light type. So they are kind of hybrid, having the properties of both heavy and light type ferritins. And the phenotype shown by us and others, it's quite impressive. So, and especially for ferritin 2. So it led us to also try, and others to try the vaccinate against recombinant using the recombinant ferritin. In some cases, it brought promising results. Yeah, and was still in interest in development of some anti-tick vaccine.
I would like to make a comment here, José speaking. It's interesting that when ticks feed on blood, there is a lot of host proteins that are secreted in the saliva. And ferritin is one of them. So, and ferritin is abundant in the blood of a vertebrate. So I wonder if there is some crosstalk between the host, and so if it's secreted in the saliva, it means that the ferritin goes through the midgut, to the hemolymph, to the salivary gland. So it's mixed in the whole system of the tick. So could it be that the transferring from the host transfers the iron to the ferritin from the tick? Because as you said, the iron, reduced iron, is only from ferritin, not from hemoglobin. So,
and I said that the iron is from transferrin, not from the serum transferrin, not serum ferritin.
oh, sorry, I confused.
Yeah, but maybe one idea was that, well, John Locke's hypothesis about transferrin was that transferrin or ferritin, the plasma ferritin, should be an antibacterial protein, because a lot of pathogens look for iron, because it's a micronutrient that's not very abundant. So, and then there is a kind of a fight between hosts and pathogens for iron, and ferritin. This was John Law's idea. Was a kind of a mop for iron. Maybe this is important for the tick, because it keeps a lot of time in the bite site. So it's amazing, because the bite site doesn't get any infection. So it's clean.
Yeah, this is the if I'm not mistaken, I think this immunity function of the iron withholding, I think it was for insect transferrin and not for ferritin.
Yeah.
So
You are right.
This is what actually came to my mind when we were writing this review. And seems to me to be very interesting, this iron withholding strategy, because as maybe I can show later, the tick midgutis and Melina knows very well that's almost sterile. So, but so we think that this may be also one of the possibilities why there are not that many microbes in a midgut, because they just do not have enough iron. But interestingly, the spirochetes, the Borrelia spirochete, they are not dependent on iron. There are some studies, so So this is maybe why spirochetes can easily survive compared to the other bacteria. But we are just very at the phase of thinking about these things, and it would be very nice to look at it closer. This is just the summary show. This is the picture we use for the review, very simplified way, saying that actually what we think that heme transport and iron transport in ticks is it's actually distributed are independent process in that tissue distribution. So iron is transported by ferritin. Iron comes from transferrin, get intracellular to ferritin 1 and transported by ferritin 2. Help is going to either with hem is going either with help to at all stages or using the vitalogenin going to ovaries. So this is kind of simplified overview of the story. There are still many interesting things which needs to be focused on this. For instance, there are felin leukemia virus, another potential another heme transporter which needs to be functional studied in ticks. For iron, there is definitely some demetal transporter. We only touch these things very briefly, and there should be more effort to focus on these other molecules possibly involved in these processes.
Petr, can I ask about vitellogenins in insects? Because I did not know that vitellogenins traffic heme. I'm embarrassed to say this. I'm glad I'm hearing this, but obviously they are very important proteins for making eggs in insects, and normally people link them with lipid metabolism. And now I did a PubMed search while you were talking, and I saw your paper and Pedro's paper, etc., and said how I had missed this. But it's all in ticks. So the question is, do insect vitellogenins bind heme? Yes or no?
I guess no. I think that they do not need to distribute heme like that because they can synthesize in ovary. So I think I have no idea that or no knowledge about that the insect like vitellogenins will transport heme, but maybe
I guess they do bind, but it's interesting because at least in the both micropus, the vitellogenin, meaning the hemolymph form, was not colored at all. But the vitelin, the egg form, was deeply brown. So, meaning from the hemolymph to the egg, it catches him somehow. Something changed.
But do you have information about Rodnius or other insect vitellogenins?
No. There in other insects, it's completely colorless.
Petro, you were talking now about Rodnius.
Yes.
Yes. Okay, I thought you are talking
in the Rodnius, the vitelin and vitellogenin, both, they are colorless. Right. But if you isolate the yolk platelet, meaning the organelle, that's pink. But because of another protein, that the Rodnius heme binding protein, that's there in the same intracellular compartment.
Mm-hmm.
Okay, so this we cannot generalize. This is a tick-specific function for the vitellogenins.
I think it is for the vitellogenin, yes.
Yeah.
Okay.
As long as I know, it's the only group that has. And all chick eggs are brown.
Mhm. So this is the story inspired by Pedro's work again, and we try to just find in ticks. This is about tyrosine degradation and actually about the toxicity of not proteins toxicity, but when they actually either silence the degradation by RNA or inhibitors of these HPPD enzymes, by like mesotrione, nitisinone, they see that it has a really little effect on rodnios, mosquito, ticks as well. So we try to repeat this for ticks, also for nymphs, and so the same. And the main question is, what's the reason for this lethality? So in rodnios, Pedro reported that it's formation of some tyrosine crystals in hemolymph, in tissues, and but we didn't see anything like that in ticks. So we are trying now. Matěj Kučera is actually trying to do, in collaboration with some group, some metabolomic studies, what happens to with the excess of tyrosine when it's actually its degradation is impaired. And there are some interesting results that it should have an effect on when tyrosine increased, there is some increased formation of melanin and sclerotization of hemolymph. So these are the slides of cuticle from ticks effects on control on mesothelioma, and we see that there are more melanin in these, and it has he did a funny experiments. I like those on the physical properties of the tick. So this is the experiment we did. We put ticks under the beaker and filled with the water and compared how much it will what will be the weight of the weight before the tick explodes. So the normal ticks they need almost 1.5 kilogram. While these were more these mesotrione were more sensible and more, I would say, fragile, more fragile. And similar experiment was done, although I don't know how they did it. This collaboration, so they measured the elasticity of the cuticle, and also found that the elasticity of the cuticle in mesotrione of treated ticks is much lower. So, but this needs to be finished. I hope that Matěj Kučera will finish his PhD and will publish this soon, I hope. This is another experiment done and another project done together with Jose Ribeiro, and the question was what is behind the actually the acquired resistance like rabbits have against repeated tick feeding. So they prepared by repeated feeding resistant ticks, which resistant ticks, which resistant rabbits, sorry. When the feeding of ticks was much less successful. Interestingly, they took the sera from these control ticks and from these resistant ticks, and when they feed them ticks on the membrane, they didn't see any difference. So it's not only in a sera. So other question was they wanted to find the antigens in tick saliva, which may be responsible for this resistance, and using the proteomic approach and affinity binding on immunoglobulins from serum, and most of those things which came out of this study were metalloproteases. And it was also kind of confirmed that when we injected a metalloprotease inhibitor, I think it was phenanthroline, orthophenanthroline. So it was quite toxic for ticks when fed on living animal, but not toxic except this high concentration on membrane. So there, it's not only the interaction of the antigens present in a saliva and antibodies present in a serum, but there should be something in between, maybe some cell immunity and whatever, which is actually responsible for the quiet resistance. And here I just briefly will go through this. We are inspired now. Up to here, it was mainly about the differences of the between the parasite and host, the major differences, and when it's kind of basic strategy to find the biggest differences between the parasite and host and try to focus on those. But now there are strategies in parasitic research that people look at actually quite similar things which has a host and the parasite and quite conserved. So, for instance, these basic enzymes like tRNA amino acid tRNA synthetases were shown that in several parasites that can be actually effectively treated by drugs, like the Plasmodium falciparum can be treated by cladosporine, and it inhibits the lysyl tRNA synthetase, or this compound lysyl tRNA, and it was shown in nice papers to function nicely against different parasites. We didn't succeed to make this with artificial feeding. Cladosporine is simply too expensive to try, and this compound was not effective, either not stable. But the effect of RNA silencing on this is actually striking. So we see that silencing of selected tRNA synthesis has a detrimental effect on tick feeding, but it doesn't kill the tick. It's important to say on adult and as well as on nymphs. And when we saw this on nymphs, the question was, will these nymphs will be able to when infected with Borrelia, will still be able to transmit the Lyme disease to the host. So there was a bet in the lab for this keg of beer, and yes, the guys used the transmission models we have now for Ixodes ricinus and Borrelia afzelii, and were able to show that these, we call them zombie ticks, are still able to infect the murine tissues with the spirochetes. So we see that it was another piece of evidence for us that we now strongly believe that, at least for this European model, we think to the original idea of Dr. Budorfer, who came up with the Borrelia spirochetes, was that the kind of direct road from the midgut to back to the host. He suggested a process like a regurgitation. We think it's active move of spirochetes from midgut midgut to back to the mouse by infecting host and actually avoiding the saliva, which is believed for years now to be the main transmission route, but we never saw Borrelia in the saliva like here, and saw them be heavily present in infected midgut. So this is we don't know yet whether this is just the specific case for Ricinus and Borrelia afzelii, because we don't have scapularis and Borrelia burgdorferi sensu stricto to repeat that. But I think this original idea should be also of Burgdorfer, this mouse, we call it gut to mouse transmission route, should be reinvestigated. And other interesting things, we just playing around. There are certainly here are the glutamate gated channels, which are targeted by the chemicals very effectively, fipronil, ivermectin. Now with the isosazolines, yes, like chloralaro. And we found an interesting article for mosquito that people are trying, actually came with the idea that to produce antibodies against the extracellular region of this glutamate gated region. So we are now trying to do the same. So actually there are two glutamate gated chloride channels we found, and mainly the number one. So we produce the antibodies against the extracellular region, and looks that immunizing guinea pigs with this has some effect. Maybe not too strong, but still seems to be kind of proof of concept that this may work. And another thing which is now kind of very interesting that for
sorry, Petra, what happens if you give insecticide to those antibody treated?
You mean for the mosquito?
What?
You mean what happens to the mosquito when they tried?
Yeah, to the ixodes if they immunized guinea pigs, and then if you add the insecticide towards the same channel.
You expect it to be protected?
Maybe.
Yeah.
That's the question.
You mean the intracellular site, or I'm not now sure whether I got it.
Well,
question.
Just apply the give ivermectin in a sublethal or lethal doses, 2 doses, and see what happens.
Mm-hmm.
If you give it to a 50% lethal dose, then you will see up or down effects of immunization.
So you mean combining some low doses of ivermectin with the antibodies, right? Targeting both.
Yeah.
Like going here, we have ivermectin. Let's say
Yes.
at this 2.5 nanomole concentration, and or a little bit less, maybe, and
something in the middle.
It's interesting.
What would be the interaction between the antibody and the antibody to a receptor that is targeted by insecticide, acaricide?
Yeah.
Is there a talk between them? It would be interesting to know.
Mm-hmm. Yeah, that's okay. It's worth to try. I regret I forgot to tell Jan Perner, because I think he may add much more to this discussion. because he's very much in this. But I'm afraid he is at home already. Yeah, this is just another thing which is now really searched by different teams. The proteasome, inhibiting the proteasome, very conserved thing in host and parasites. And the question is just to find a good therapeutic window for a compound which will not too much harm the host and will harm the parasites. So was found to be good for Plasmodium, was for other against Trypanosoma, Leishmania, and so. And Dan Sojka is now trying this for finding and testing these compounds. More of them were actually approved against Babesia. So it's really working also, and are trying with cooperation with I think San Diego to find another compounds which will be even more effective, and also they try using the artificial feeding against the ticks, and some of these compounds seems to be really toxic to the ticks, but still the concentration are not that nice. So, but the effort is to find other and better compounds which will be really specific for ticks. Also may become a promising target for chemistry. And this is just briefly the what I already mentioned, the Melina stuff. So she found that in Ricinos the microbiome in tick gut is very actually almost sterile. This the green line shows the actually noise of the analysis when you just use the distilled water, and there are huge differences in microbiome between the mid gut, very low, very unstable, no core in symbiont, and the ovaries, which is actually occupied almost exclusively with midichlorium mitochondriae in case of Ricinos. So we have the questions: what's behind the actually this protection against the microbes in tick gut? So there are several hypotheses. One, of course, is antimicrobial proteins, which are there. There is definitely the results shown earlier by actually Andrea Fogaka and Shirley Daphne, showing the hemosidin, the hemoglobin fragments. Yeah, and then the idea which came from Pedro, that the microbes can be actually endocytosed by digestive cells. Other possibilities, migration to the ovaries, and as I already mentioned, now there is also very interesting the effect of iron withholding. So the one question we solve, or Melina and we are solving now, whether the midichlorian mitochondria is really essential endosymbiont. So so far we were able to actually, using the tetracycline, we were able to make the whole development cycle of the midichlorian free ticks. So there is some impairment of the growth, but not very striking or not too striking, showing that the midichlorian is probably beneficial, but not essential. At least this is our current interpretation. Maybe Melina can say it better. So we are now working to get these data together, and regarding the antibacterial peptides, our transcriptomic and proteomic analysis actually revealed 2 mid-gut specific defensins. One we call late because it's upregulated in fully fat ticks, and another one we call early because it's present only in unfed ticks. So this called in so it's regarding this antibacterial, and Melina observes striking clearance effect. I think it was within 1 hour when they infected by capillary the ticks, either with this control gram-negative Chrysobacterium or this gram-negative Pantoea, which was isolated from ticks, or they observed completely clearance of the gram-positive Micrococcus luteus within 1 hour. So it's really striking efficiency. We just wanted to see whether really the bacteria get into the midgut. So we see that we use the green fluorescent E. coli and see that the capillary infection works well. And it was another transcriptomic project made by Melina and Jose Ribeiro, which actually shows whether there are any differences in unfed ticks, in midgut of unfed ticks when infected with these 2 Pantoea and Micrococcus. So currently this is also now processed for publication, I guess. And the 2 defensin, this is the experiment which is actually running.
One question, Petra, about the last slide, the previous.
This one?
If you add that endogenous bacteria, if you give them more than 10 to the 4, so what happens? It goes to 10 to the 3, or it just goes to a 10-fold level?
I think it was in 1 hour. It's a little bit seems to go to 10 to the 3. It's slightly reduced, but this one we know that this endogenous will be probably not completely killed, because when we did some other experiments, it's really this will kill the tick. So it will multiply that later, and the tick will die. So this is bacteria which is actually it killed our soft tick colony, and we found that it's also bacteria which can multiply and definitely kill Ricinus as well. So this is just the data for 1 hour, and we were also trying to find some non-lethal concentration of endogenous, which will not kill them, but we will not. We didn't succeed, and all concentration we tried was actually lethal for ticks. So that's why we use it to have a control that it's really going to the midgut, and yeah.
Petra, may I?
Please.
Sure. Pedro, so Pantoea is going to be eliminated, fully eliminated, as well as Micrococcus luteus by Ixodes ricinus. But what happened is that it's going to be a little bit slower than for Micrococcus luteus. So this is the results 1 hour after feeding. But if we follow the bacteria for Pantoea, if we follow the bacterial elimination for all of them is going to be eliminated in up to 24 hours. So it seems that it's not a granny specific, the elimination. It's just a little bit better, or let's say faster, for Micrococcus luteus, which is a gram-positive. It's, yeah, that's it.
And true is that we don't have in this longer intervals for endogenous. That's right. Because it was just used here for as a control for 1 hour exposure. And as I talked before about these two defensins, we get them synthesized. The full defensins are mature defensins in China, and including the disulfide bridges. And we tested them in these growth curves. And actually, one of these experiments just running overnight now. And we found that these both defenses are extremely potent against this Micrococcus luteus and against some other gram-positive like Staphylococcus aureus and Staphylococcus epidermidis, but not at all against the gram-negative. And now today we are just testing against these two defensins against the Chrysobacterium, against the Pantoea, and against lower concentration of Micrococcus to really get the IC50 more precisely. And also we are trying to in the experiment is we reduce the disulfide bridges of these defenses to see whether they are important for the activity. So this is the experiment which is running this week. So I'm really excited to see the results. And I guess it's the last experimental paper. So this is the lab from summer 19, including Melina here. This is Honza Perner, the current head of the lab, and Dan Sojka, you know him, and some others. And now the lab is extended for the group of Ondra Hajdušek I don't have here. And here is the acknowledgements to the grant agencies and to the people working in the lab, and definitely the students. And I hope you don't mind that the whole talk was about how to kill the tick. But so, if there are any questions more than to me, Lina.
I have a very small technical question, because I saw that you used to stain the lipid droplets with the LD540.
Yeah.
And I looked before to buy it and I cannot find. I am about to write to the people in Germany to get some. So how did you get some?
Yeah, it's there was a paper. They get it synthesized by some company, and de novo synthesis is very expensive. But we acquired this company, and because they have already like the recipes ready, so we got it for relatively reasonable price from this company. And I'm not able to find it now. What was the company? But definitely I would be able to.
Okay, I think I will write to these people in Germany that described the compound. Maybe, yeah.
Yeah, but we then contacted the company they got it from. And they offered us the same compound for a really reasonable price.
Ah,
and it's working very nice for the lipid droplets.
Yeah.
We have a question in the chat by Jessica Fiorotti.
Yeah. I think it's better, Petra, close the presentation.
Yeah, okay.
Okay.
So
Sorry, Pedro.
Yeah.
How to do it now? Just close it.
Just
Mhm.
stop sharing.
Yeah,
Yeah.
perfect. Thank you.
Then you see her question.
Now I see the audience.
Yeah.
Oh, Jessica. I didn't mention the collaboration with Jessica. It was another project, nice project on entomopathogenic fungi, but much more related to tick immunity. So it was completely. It was not involved in this presentation, and honestly, I feel sometimes that we really are hunting at the one moment too many rabbits, and we should know what is the most promising, but we still don't know where the golden apples are. So that's why we are so topically so spread it and unfocused. And
If you knew the answer, you should work in Wall Street.
good point.
Questions?
I have one from the beginning of the talk when I was too shy to interrupt. When you put the proteomic slide of what was there in the midgut, and did I understand correctly that you find so my question is, what do we think? Are there some proteins that the tick feeds on and they are not degraded and they can move inside the tick without being degraded in the midgut? That's the question.
I think it's very good point, and especially, I mean, it's especially important for, there are so many, especially for immunoglobulins of the host origin, and also there are strategies, anti-tick vaccine strategies, and many of these things are focused on not only on things expressed in the midgut, but on things which are present on other tissues or in hemolymph. And there are data, and we can, I think, we can see some immunoglobulins of the guinea pigs. We also have some not only midgut proteome, but the hemolymph proteome, and we see some.
But has anybody ever proposed a way by which proteins escape the various proteases and get integrated inside an intestine? Because I have seen here and there evidence that this happens, but I have never read something how it happens, you see, and then it is a little bit in contrast to what we teach happens during digestion, right? So for me it's a big question, and
it was the digestive system machinery.
Pedro has some findings of albumin transport to the midgut. Can you say that, Pedro?
What?
You have shown that the transport of albumin to the midgut and hemoglobin, that there is a kind of a shuttle for proteins into the midgut.
Yeah, well, the idea was that hemoglobin was recognized by a receptor, but albumin went into a different compartment, probably by means of fluid phase endocytosis. But that inside the digest cells. So the fact is that Well, the first paper we published together with ITA, LOTA, 1986, was an evaluation of the amount of immunoglobulins that were found from cattle plasma. And about 5%, if I don't...
1%, 1-2%.
1-2% of the immunoglobulins were found in the hemolymph. So it was a kind of, the guy was leaky to the, so a large amount of
saliva. It leads to the saliva. The saliva, you find the hemoglobin, you find everything.
Yeah. So it's a continuum between the host and the inside of the tick. Almost the same guy.
I wonder if in the saliva, the salivary gland, if the proteins can pass through the Golgi or endoplasmic reticulum, if they can be glycosylated with the glycosylation pattern of the tick and then injected back into the host. And these would create the perfect immunosuppressive system, because you would have at the same time self proteins, peptides, and then the glycosylation pattern. And usually, glycosylation patterns of insects are highly immunogenic. So that's my speculation.
Yes, it's happening, but nobody knows how it's happening. So.
But this is a good point to really look at our proteome analysis of hemolymph and to look for the host proteins.
But if you do the proteomic analysis and it's glycosylated, that would be just as the non-identified fragments, because the molecular weight would not match anything.
Yeah.
But when you do the tryptic digests, you only, if you find 5 tryptic digests that have no glycosylation pattern and it matches something, it's a positive.
It's a positive, but then you find, but that would not help with to test the hypothesis of glycosylation.
No, I think the hypothesis of glycosylation, you have to do maybe to find some lectins that could bind. If you could purify the hemoglobin that is coming out in saliva and purify it and see the lectin binding pattern that they could have, I would have to find some glycobiology lab to interact, because it's another world.
Yeah. Yeah, maybe a G column, Anderson, or maybe concanavalin A.
Yeah, actually, Con-A binds very well to Hymenose type.
Yes.
Hymenose is very foreign to us, because we normally have the Hymenose are the basis for making the complex carbohydrates. Actually, the ovalbumin has a lot of Hymenose, and it's the main antigen from ovalbumin to guinea pigs and humans. And it binds Con-A very well. But I don't know what are the tick glycosylation pattern. Actually, you probably have found the alpha-gal story that it has induces anaphylactics in humans. So there is some interest in studying the glycosylation pattern, but I don't know.
Yeah.
It's, I think, a mature area for somebody to grab.
Another area to focus on.
Yeah. Well. Okay. Sounds good. So I think it's, we have a. I want to make a question, a personal question for José. Zé, you stand up for 2 hours now. So that's unusual. It was because the conference was specifically interesting, or you are getting better also?
It was the time. I get about 3-4 hours in the morning and about 3-4 hours in the afternoon. And I have to lay down between noon and 4 o'clock. So it was perfect because I got in my second. But I'm getting a little better, but not yet close to be fully. My battery is not recharging.
Well, small steps.
Yeah.
Great.
But was a great talk, Peter. Thank you very much.
You should know that I'm not able to present clearly anything, so there are much better people to explain things. Next time save Jan Perner, he will be more specific, I guess.
Send him my regards.
Yeah, for sure.
Yeah, but we have a very nice couple of hours.
Yeah.
It was great to see you again.
It was a real pleasure to see you at least like that, So
Thank you a lot, Petra.
thank you.
Congratulations.
Ciao, ciao.
Thank you. Bye-bye, guys.
Bye-bye.
Bye, everybody.
See you
Tchau,
later.
gente. Bom
Tchau,
carnaval
tchau.
e resto de carnaval.
Oi, Anderson. Um abraço.
Tchau, tchau.
Até
Bye.
mais, abraço, Zé.
Tchau, Ita.
Ciao.
Tchau, Zé. Abração, Zé.
Tchau, Pedro.
Bye.
Tchau, Zé.
Bye.